Good morning, everybody. So, welcome all of you on our YouTube platform, Genomax. Okay. So, today we are going to talk about some of the miscellaneous topic of our genomics. That is, and it is also related to immunology. Okay, everyone. So, today we are going to talk about the hybridoma technique, as you can see here. Okay, everyone. So, in 1975, Kohler and Milstein first fused lymphocyte to produce a cell line which has both immortal and a producer of specific antibodies. So, friends, before 1975, we are able to produce antibodies, but they are not specific only for a single antigen. But in 1975, Kohler and Milstein first fused a lymphocyte. But which type of lymphocyte? Basically, it is a B cell, because we all know that B cell are able to produce antibodies. Okay, that's why Kohler and Milstein fuses a lymphocyte with a tumorous cell which is known as myeloma cells and created a new cell line which by which technique this technique is called hybridoma technique. And in hybridoma technique we create a new cell line which is specific for a single antigen. Okay, everyone. The two scientists were awarded the Nobel Prize for Medicine in 1984 for the development of this hybridoma. Because of the formation and because of the invention and discovery of this technique, both the scientists had awarded with Nobel Prize in 1984. The value of hybridomas to be filled was not truly appreciated until about 1987 when monoclonal antibodies were regularly produced in rodents from diagnostic. So as we can see here, it is not truly exploit until 1987 because monoclonal antibodies at that time was produced in rodents. Okay, everyone. So we talk something that are apart from this is how antigens immunized and they revoke our immune system. In a specific immune response, only those B and T cells can bind to the antigens of the pathogen are selected to participate in the response. As we know that there are many types of B and T cells are present in our body, but there are only those of our who have the ability to produce immune response only to those antigens which they are specific for it. Okay, everyone. So as we can see here, mature of T and B cells with different antigen specificities, and antigen with two epitopes, red epitope and blue epitope. So as you can see here, proliferation of cells with receptors capable of binding epitopes of the antigen. ठीक? Okay, everyone. So those cells which have ability to join with the blue epitope are start proliferating, and those cells which has ability to bind with the red epitope of the antigens starts proliferating. But not all the cells start proliferation because they are specific for those epitopes which are found on the surface of the antigens. Okay, everyone. I think you get my point. Okay, everyone. Clonal selection of lymphocyte during the specific immune response. When our immune response evoke, the only a selected cells start proliferating to bind on the sides of the surface of the antigen and starts killing process and starts the process which exert and which start the killing of the antigens. Okay, now we are talking about polyclonal antibodies. If an animal is immunized with a protein, here the word immunized means we want to revoke its immune system to measure the immune response. Okay, so in immunized process we use a antigen which is basically a made up of a protein. As we see here, if an animal is immunized with a protein, a wide array of B cells will be stimulated to produce anti. As we see here, if an animal is immunized with a protein, basically the protein here is a antigen. A wide array of B cells will be stimulated to produce antibodies. Okay, everyone. So Anti-protein antibodies means the proteins which is produced in a response of those proteins which acts as a antigen. So that's why it is called anti-protein antibodies. Antibodies may be made to a number of different epitope of the protein. Okay, everyone. So as we know here, an antigen have different kind of epitopes. For a unique epitope, there is a specific antibody. So if we take an example of an antigen, it maybe have 2, 3, and 4 and 5 epitopes. It means they have a 4, 5, and 6 type of antibodies which are able to attach on it, because every epitope has its specific antibody and every antibody is specific for a epitope, vice versa. Okay, everyone. Now, even antibodies that bind to the same epitope may have different antigen binding sites. Okay, everyone. What we learn here? Every antibody that bind to the same epitope, please remind it. Okay, everyone. And we even antibodies that bind to the same epitope have different antigen binding sites and they bind the epitope with different affinity. Okay, everyone. If an antibody that is bind on the same epitope have different antigen binding sites, it means if I have a master key, it means I'm able to unlock various types of locks. Okay, everyone. But the affinity, the ability to attachment, it means there are different types of antibodies that have different affinity to attach with the epitope of the antigens. Okay, everyone. The mixture of antibodies produced in response to an antigen are referred to as polyclonal antibodies. So, okay, everyone, if I insert an antigen in an human body, it starts proliferating different types of antibodies. Okay, everyone. Then the mixture of antibodies that are start producing in our serum, it's called as polyclonal antibodies, because there are various types of antibodies that are produced in a serum, and the various types that are present in that serum are termed as polyclonal antibodies because they have large number of diversity. Okay, even they are produced by many different clones of B cell. As we know that a particular B cell are able to produce a particular antibody. Okay, everyone. So the polyclonal antibodies are produced by different types of B cells. Okay, as we can see here, when an antigen is incorporate in our body, we see they have a different type of epitopes. Okay, so what it will do, it will revoke our immune system. In in response of evocation of our immune response, the antibody starts proliferating and it starts producing in our bloodstream. So a mixture of antibodies that you can see here, a green antibody, a blue antibody, and a light green antibody, a light blue antibody, and a red antibodies are kind of different types of antibodies, and these antibodies are formed by different types of B cells. A mixture of antibodies that all bind to epitopes of the original antigen. Some bind with higher affinity than others. It means some are bind, they all have ability to bind on the surface of an antigen, but they have a different affinity. Okay, everyone. So affinity also plays a very important role in immune response. Those antibody which have higher affinity, which have higher affinity, can kill that protein, and that can help the killing of protein more better. Okay, everyone. I think you get it my point. So polyclonal antibodies. Polyclonal antibodies are a mixture of antibodies which different antigen binding sites that may bind to different epitopes or antigens of the immunizing agent with varying affinities. They may be of different antibody classes. Okay, everyone. So as we all know that there are different types of antibodies that are found in our body. That may be IgM, IgG, IgA. Okay, everyone. So polyclonal antibodies are those antibodies which have a different binding sites and which are produced by different types of B cell and which have a different affinity. Okay. Okay, let's start. So how we so the question that we are click our click in our mind, how we isolate these polyclonal antibodies. The serum obtained from an immunized animal is referred to as polyclonal antiserum because this serum consists polyclonal antibodies. Okay, everyone. A polyclonal antiserum consists antibody to different epitopes and different antigens that were present in immunizing inoculum. So, how we separate this? So, here we use a affinity chromatography. Okay, everyone. So, in affinity chromatography, you can see here, as you can see here, here is a blue matrix in which the antigens are attached on the surface of a matrix here, and the pink one are a matrix, and blue, the small blue dots are specify the antigen, and here the red dots represent the antibody specific to the antigens, and here the brown one represent the known antibody protein. So here is the way. So we use affinity chromatography to isolate the various types of antigens, various types of antibodies from our antiserum protein. Basically these are polyclonal antibodies. Okay, everyone. Now we talk about hybridoma techniques. ठीक? B lymphocyte can mutate into tumor cells that result in a type of cancer termed myeloma. Okay, everyone. So as we know that B lymphocytes are not the immortal cells. They have a definite lifespan. After that, they will die. Okay, everyone. So myeloma cells become immortal and will grow indefinitely in culture. So myeloma cells basically are the cancerous cells, and they have ability to not die because they have a specific type of cell growth, and this specific type of protein which never stops its cell cycle, and they will never gonna die. So but B cells have a different lifespan. So what we do a fusion. Okay, we do a fusion between a B cells and the myeloma cells, and what we find a new combination of a cell line which have a ability to starts and which have ability to keep producing the antibody, only a one type of antibody, because we are using only one type of B cell here, and that B cell have a immortal capacity. So it starts producing antibody, but they will never die because it is a fuse with a myeloma cells. So how we do it? So you can see here, we isolate a specific V cell from a body, and then we growth with then we fuse it with a myeloma cell, and we use a special media which is known as HGPRT media. Why we use HGPRT media? We use HGPRT media because it separates the non-fusionable cells with the fusionable cells. Those cells which are not fuses with the myeloma cells, it will gonna die, but those cells which are fused with myeloma cells, it starts producing antibody and it's never gonna die. So hybridoma cells grow indefinitely in cell culture and secrete antibody, and after we after the selection, we exert our monoclonal antibodies, and we can use it for our another processes. So here, monoclonal antibodies produced from a single clone of B cells, producing by fusing a B cell secreting the desired antibody with the myeloma cell, capable of growing indefinitely in tissue culture. Monoclonal antibody all have identical and antigen binding site, thus they all bind to a same epitope with the same affinity. They are all same antibody class isotype. So why we want and why we need of a monoclonal antibodies? We use monoclonal antibodies in diagnostic processes, in diagnostic process in that such as in ELISA, and we also use in medical purposes, and we also use on treatment processes. Okay, because they have a specific site which is bind with the specific antigen. Okay, everyone, so that's all for today. I hope you like this video. If you like, please like, share and comment. If you want the application of the hybridoma technology, please do comment on the comment box. So that's all for today. If you like this video, please like, share and comment, and don't forget to subscribe, and please hit the bell icon for the updation of the next videos. Thank you everyone.